In this lecture, we'll be focusing on the genetic basis of development. So we'll talk a little bit about the process of development and then the information that's needed to guide this process. So in the very beginning, there's the process of fertilization where egg and sperm come together to produce a diploid zygote. The zygote then must develop into a multicellular organism. There are three key processes that occur during development that are going to aid in the zygote development so that we have a fully formed multicellular organism at the end of this process. One is cell division, which is one of the first things to occur after fertilization. The zygote starts dividing to produce a ball of identical cells. These are all stem cells, they're undifferentiated, and they're all identical in genetics as well as structure and function initially. Then the cells differentiate later on. So differentiation allows for the cells to specialize in structure and function. The genome in each of those cells remains the same, but it's the epigenome that differs, the portion of the genome that's being expressed in each of those cells. And then lastly, we have morphogenesis, where the cells are organized in a certain way into tissues and organs to produce the correct three-dimensional arrangement of structures in the organism so that everything's in the right place. Differentiation involves differential gene expression, meaning that genes are regulated differently in each of the cell types in the body. So we have different sets of genes being expressed in these various types of cells, or each of these types of cells has a unique epigenome. This diagram over here does a good job of showing some of the process of embryonic development. So here we have egg and sperm coming together in fertilization. That will produce a zygote that then divides by mitosis to produce a ball of cells, which is known as the morula. These are undifferentiated totipotent stem cells, meaning that each of those stem cells has the potential to become anything in the body. The cells then rearrange themselves based on information that they're receiving into various layers. So there's an outer layer of cells and an inner layer of cells, and these cells are now destined to become different parts of the body. So the inner layer of cells that's highlighted here, the blue ones, they're referred to as pluripotent stem cells. They're no longer totipotent because that inner mass of cells is destined to become only a portion of the body, and the outer layer of cells will become other components in the body. So pluripotent stem cells are somewhat limited in what they can become, but they can still differentiate into a number of different cells. Let's go back for one moment. One thing that I forgot to mention, the miRNA that plays a role in gene silencing. So we talked about this in the context of regulating gene expression, but miRNA is one of the small RNA molecules involved in RNA interference, or RNAi. So miRNA has been found to play a key role in gene silencing during the process of embryonic development that is going to permanently turn off certain genes in different cells. So something like a nerve cell does not need to be expressing genes that are specific to a muscle cell, and so those would be permanently turned off, and miRNA plays a role in that. So we'll talk about a couple of different sources of information that help to guide the process of cell differentiation. So cells need to obtain information so that they know what they need to become. So one form of that information are cytoplasmic determinants. Cytoplasmic determinants are molecules that are in the unfertilized egg cell. So this is the female gamete, and when those gametes are forming in the female gonad, so in the adult female, there are molecules that are produced by mom's cells and then put into the egg cell. And they're unevenly distributed. So this could be collections of mRNA molecules that are transcribed from mom's DNA, proteins that are produced by mom's cells, other substances that are being incorporated, maybe they're ion concentrations, whatever it might be. Even the organelles may be unevenly distributed so that certain cells have higher numbers of a particular organelle over others. All of this is unevenly distributed. So there are essentially these concentration gradients of these molecules in the egg cell. And these molecules can influence the course of early embryonic development. It's a form of information that's going to tell those embryonic cells what to become and where to go in the body. After fertilization occurs, the zygote that also has all of these cytoplasmic determinants in there in uneven distributions goes through mitotic divisions. And when that zygote divides by mitosis, the daughter cells wind up with unique collections of these cytoplasmic determinants. One daughter cell is a little bit different from the other daughter cell in the concentration of these molecules that they have. So in the diagram over here, here's the unfertilized egg cell, and we're just focusing on two cytoplasmic determinants, the orange circles and the green triangles. So you notice that there's a high concentration of orange circles on the left-hand side of the egg cell and a high concentration of green triangles on the right side. So after fertilization occurs, those concentration gradients remain relatively the same, and then the zygote starts dividing by mitosis. So when the nucleus is duplicated and the cell splits in half during cytokinesis, you wind up with two daughter cells that are genetically identical. Under the microscope, they look exactly the same. So in terms of structure and function, nothing has really changed, but the collection of information molecules in each of these cells is different. And so in the course of embryonic development, these two cells and the daughter cells that are the result of these two cells will ultimately take two different paths to become different parts of the body. So the mitotic divisions that produce the daughter cells, in the context of looking at the cytoplasmic determinants, it creates daughter cells with unique collections of cytoplasmic determinants. Different sets of information will lead to different products in differentiation and morphogenesis. Another source of information is induction. So this has to do with signaling from other embryonic cells. So this is not information from prior to fertilization and development, and it's not information from the outside. This is information from one embryonic cell that's influencing the development of another embryonic cell. So based on the position, or maybe based on the cytoplasmic determinants that are in some of these embryonic cells, it causes them to produce signaling molecules, which are known as inducers, that are released and will influence other cells in the embryo. So those signal molecules bind to receptors that leads to a signal transduction pathway that ultimately changes gene expression in the recipient cell, and so it will change the path that they're on in terms of differentiation and development. Lastly, there's communication between neighboring cells. This can be through direct contact, maybe molecules on the outside of one cell are interacting with receptors on an adjacent cell, and so they're communicating their position sometimes. Where am I in the embryo? And there are some cells that will be directed to produce growth factors that will be released. Remember that growth factors are a form of paracrine signaling. It remains within the local region. It's not going to go body or embryo wide necessarily, so it's going to influence those neighboring cells right in that area. The process of determination is essentially what leads to differentiation. So in determination, the cells are receiving a whole bunch of information that's going to ultimately determine what it becomes at the end of differentiation. Determination is an irreversible process, so the information that the cell is receiving is putting it on a path that leads to a specific type of cell, and there's little we can do to change that path once that information is there. So cells that are moved to another location, let's say this is a cell that has received a whole bunch of information that tells it that ultimately it's going to become a muscle cell. It hasn't become a muscle cell yet, but it has all this information saying that it will become a muscle cell. If we take that cell and we move it into a region in the embryo that is full of cells that are going to become nerve cells, that cell still becomes a muscle cell because it received all this information that is changing its gene expression, and the genes that wind up being expressed say muscle cell. Determination ultimately leads to differentiation. So all that information that was collected is guiding the gene expression in that cell so that it's expressing cell-specific proteins. So in differentiation, cells specialize in structure and function because of the expression of genes that are tissue specific or specific for a particular type of cell. Those are the proteins that are being produced, and that's what makes it a muscle cell, or that's what makes it a nerve cell or a skin cell, etc. Stem cells are the undifferentiated or unspecialized cells that ultimately become specific types of cells. They can become more specialized. In this diagram, we're looking at the process of determination and differentiation for a muscle cell. So at the top, we're starting out with an undifferentiated embryonic stem cell, and in that stem cell, the genes that are specific to muscle cells are all off. Then that embryonic stem cell receives information, which causes it to turn on gene expression of this master regulatory gene, MyoD. Now, in terms of structure and function, this still looks just like a stem cell. There's been no change in structure and function, but we've had this single change in gene expression. This codes for a transcription factor, MyoD. MyoD is then going to regulate gene expression of these muscle-specific genes over here and turn on gene expression, which codes for this transcription factor, and then the transcription factor turns on these genes over here, which is going to lead to the production of proteins that are specific to muscle cells. So this process here, up here, is determination, where the cell receives information that sets it on a path to become a specific type of cell that cannot be changed. Once it's set on that path, it will become a muscle cell, regardless of what else happens. And then in the process of differentiation, that stem cell starts producing cell-specific proteins, muscle-specific proteins in this case, and then it differentiates into a muscle cell.